Good morning, everyone. I'm Onesme. I'm pursuing course called Biotechnology in Master. So today I'm presenting my assignment on Manovis automated protein annotation, basically in creation in pipelines in protein database. So this topic is extremely important in modern bioinformatics because nowadays biological data are growing rapidly and scientists need efficient ways to annotate protein sequence accurately. So in this presentation, I will be mainly explaining manual annotation, automated annotation, hybrid pipelines, and AI integration, and even future training in computational proteomics. So this is our outline or introduction, how can I say. So it is divided mainly into 7 sections. First, I will discuss information and importance of protein annotation. Then I will be explaining the major protein database and manual method, automated annotation system, comparative analysis, and hybrid pipelines. And even finally, we talk about future development, including AI and alpha code. So we are currently facing the major protein annotation crisis. Modern sequencing technology generates millions of new protein sequences every year, but our ability to interpolate these sequences is much slower. Without a proper annotation, protein sequences are simply chain of amino acid with no biological meaning. So protein annotation converts those raw sequence data into variable sequence knowledge. If you look at this slide, it highlights the scale of problem. So the genomic evolution has created an enormous amount of biological data every year. Millions of new protein sequences are added to the database. Annotation is important because it supports drug discovery, disease research, evolutionary biology, and precision medicine. Without annotation, these sequences cannot contribute to biomedical research. Protein databases are the backbone of bioinformatics, as everyone knows. Different databases store different levels of information. So some databases contain low sequence while others provide created functional and structured data. These data works together to support protein annotation pipelines. There are three major categories of protein database. The first is the primary database like UniProt, TrEMBL, and Genomics, which store raw protein sequences. The second is databases like Swiss-Prot and InterPro, which provide expert created annotation. The third is structured database like PDB, which contains three-dimensional protein structure important for functional prediction. The human touch. So this is manual annotation depends on expert biocurators. Scientists carefully analyze experimental evidence, research papers, and protein structure before assigning annotation. This process is highly reliable and biological accurate because every annotation is manually validated. This slide explains the Swiss-Prot manual annotation workflow. First, the protein sequence are verified for quality and duplicate are removed. So next, curators analyze scientific literature to collect the experimental evidence. Then functional terms and ontology classification are assigned. Finally, the data undergoes quality control and expert review before publication. So Swiss-Prot is considered the gold standard of protein annotation. A genomic data expanded rapidly. Automated annotation system became essential. Automated pipeline are computational tools and AI algorithms to process huge numbers of protein quickly. This system provides speed and scalability that manual methods alone cannot achieve. There are four major computational approaches in automated annotation. First is homology-based annotation using tools like BLAST, HEMA. Second is domain-based prediction using Perform and InterPro. The third is machine learning, where AI model predict protein function from patterns in data. So finally, AI structure prediction tools like AlphaFold predict three-dimensional protein structure directly from sequences. This slide compare manual and automated annotation systems. So as we are seeing this slide, manual annotation provide high accuracy, but often above 95%, but it's slow and expensive. So automated system process millions of proteins daily and provide massive coverage, but accuracy may be lower due to prediction errors. The future of protein annotation is hybrid equation. Hybrid systems combine machine efficient with human expertise. Automated tools perform large-scale prediction, while human expert evaluate difficult or important cases. This approach balances scalability with reliable. This slide explains how hybrid pipelines work in databases like UniProt and InterProScan. So first, automated pipelines analyze all protein sequences and assign confidence scores. Low confidence or biological important data that then reviewed by expert curators. So validated information is fed back into AI training system, continuously improving future prediction. Despite major progress, several options remain. So sequencing data is growing faster than annotation system can handle error propagation and speed incorrect annotation across databases. Many proteins we have unknown functions and no homology. Emerging solutions including AlphaFold, protein language models, metagenomics integration, and cloud computing technology. By concluding, in conclusion, manually annotation provides high accuracy while automated annotation provides scalability and speed. The most effective strategy is hybrid creation, which combine AI-driven prediction with expert validation. So future technology like AlphaFold and deep learning will not replace biocurators. Instead, they will enhance human expertise and improve medical research. If you have any question, drop it. Thank you.